Alright guys, welcome back to Underground Science, and in this video we'll pretty much pick off from where we left from in the last video, in the part 1 video of the synthesis and secretion of insulin, where we just talked about the overview of insulin and some effects that insulin can do to other parts of the body. Alright, and so let's go and get started. So remember, we won't get into it this time because we already said in the last video, but remember in the last video we said that insulin is secreted from our beta cells of our pancreas, right? Part of our endocrine system of our pancreas, which makes up 1% of our pancreatic cells. There are other islets of Langerhans, like delta cells, F cells, PP cells, alpha cells, and also beta cells, which we'll talk about, or which is the focus for synthesis of insulin. All right, so let's go and get started. Sorry about that. All right, so let's remember from the last video, right? We have our pancreas here, okay, and that's pretty bad drawing of the pancreas, but let's just get the idea. We said that along with our acini, right, of our pancreas, we had 1% of the pancreatic cells made up of islets of Langerhans, which we said were in clusters, right? And so let's go and draw different cells representing different islets of Langerhans. And what we're going to do in this video is we have to look at what happens inside a beta cell. All right, so let's go ahead and look at what happens inside one beta cell. All right, so let me go and draw it out. All right, here we are. So I just do the top part of a phospholipid bilayer of our beta cell. So let's go and draw this. Write this. This is our beta cell of, sorry about that. This is our beta cell of our pancreas. All right, so it's the beta cell of our pancreas. And so what happens, guys, is that we have, let's say, we have many blood vessels running by our cells, right? So let's take a blood vessel right here. Let's say we just ate a meal. So we're going to have high blood glucose levels, right? We're going to have high blood glucose levels, and we need insulin to bring those blood glucose levels down. Well, what's going to happen is that, and remember, guys, remember our GLUT4 receptors are insulin dependent. They need insulin in order for glucose to come into them. That's why insulin brings blood glucose levels down. All right, and so what's going to happen is that our beta cells are the cells that's producing the insulin. So it won't make sense that our beta cells are insulin dependent because they're the one who's producing it. So we're actually going to have not GLUT4 channels or GLUT4 protein transporters. We're going to have GLUT2. So let me go and draw that out. So we're going to have a protein channel on our cell membrane of our beta cells. It's going to be a GLUT2, a glucose transporter type 2. One thing to know about here is that the GLUT2 transporter is bidirectional. All right, so it can go both ways. So if glucose comes in, it can go out. All right, but we'll see how it can't, how our cell intelligently makes it not go out. So then glucose will diffuse from high to low concentration, and it's going to be facilitated through this GLUT2 transporter. So this GLUT2 transporter is going to help glucose come into the pancreatic beta cell. All right, so now we're going to have glucose inside here, inside our pancreatic beta cell. All right, so remember, this is the inside of the pancreatic beta cell, and this is the outside of the pancreatic beta cell. Now we have glucose inside. So before we start talking more about this, let's talk about how insulin in the first place is being made inside the cell. So we have our nucleus, right, inside our cell. Then remember, DNA is tightly wound through histone proteins, and we have chromosomes, right, which is another way of packing that DNA even more into a smaller area, right? So let's say we have a chromosome in our nucleus, right? We have chromosomes. However, in chromosome 11 is where, in general, our insulin protein synthesis takes place. All right, so that's where we transcribe and translate a protein, or that protein will be insulin. So in here, we have all our DNA, right? We have all our DNA here inside our chromosomes. So if we zoom out, we'll see actual DNA tightly wound if we exclude the histone proteins for now, right? And in chromosome number 11 in particular, right, what they saw was that in chromosome number 11 is where most of the insulin synthesis occurs. all right, because the chromosome 11 has genes on its DNA that will actually, through transcription, we will eventually get a mRNA strand, right? We'll eventually get an mRNA strand that will eventually go into the cytoplasm. So remember, transcription occurs in the nucleus. So just to be more accurate, sorry about that, let me not draw it like this. So what happens is that mRNA is created, right, or synthesized inside the nucleus. So we're going to have mRNA here. Right? First, we're going to have pre-mRNA, but we'll go through protein synthesis in a future playlist. So let's just say we have mRNA here. All right. I'm going to draw it, write it in black so you can see it better. And then mRNA now from the nucleus will actually exit and go to the cytoplasm of the cell. And we'll find a large ribosomal subunit and a small ribosomal subunit. All right. And this mRNA strand, right, can then undergo translation, right, through the help of tRNAs and codon and anticodon pairing, and generate a polypeptide, and we'll synthesize a polypeptide. So from here, we'll actually synthesize a polypeptide. Now this polypeptide isn't insulin yet, all right? This polypeptide will actually have to go first to the endoplasmic reticulum of our beta cell, all right? So we'll have to go to the endoplasmic reticulum first. When it goes to the endoplasmic reticulum, it's actually going to be converted into a structure known as proinsulin. All right, so it's not insulin yet, but it comes out as proinsulin. All right, so it comes out as proinsulin. All right, so proinsulin will then go to the Golgi apparatus inside our cytoplasm of our beta cell, right? So let me go and erase that. I was kind of distracted by that. All right, and then it's going to go to the Golgi apparatus. Golgi, oops, that's not how you spell it. All right, it'll go to the Golgi apparatus. And once it goes to the Golgi apparatus, guys, it's going to get packaged into insulin. So once it comes out, it's going to get packaged all over, right? And it's going to come out in a vesicle. So let's say we have this huge vesicle here. In this vesicle, we're going to have zinc. All right, we're going to have zinc. And then we're going to have a few insulin, protein, peptides, right? Insulin peptide hormones. So let's just go and draw insulin here, right? Let's draw one, all right? But we know there's actually a good amount of insulin in each vesicle. All right, so I think there's actually 6 insulin per zinc, right, in each vesicle. But let's just go and draw one insulin here. So let's go and write down insulin. All right, and actually, you know what? To be more accurate, we should draw it bound to zinc, all right, because it forms a hexamer. All right, so six insulin molecules, right, is bound to a zinc inside our vesicle that came at the Golgi apparatus. So let's go and draw, let's do one big one, right, representing insulin. And then let's represent 5 smaller ones, right, but it's still insulin. So 1, 2, 3, 4, and then 5, and then 6. So that's 6 insulin bound to the zinc. And also in this vesicle, so we have insulin and zinc so far, right? We also will have another peptide, all right? It's going to be called the C peptide, all right? A C peptide, and we're also going to have a little bit of amylin, all right? A little bit of amylin, which can lead to bad things because it can actually lead to the destruction of your beta cells. It can actually damage your beta cells, I should say, if we secrete too much insulin, all right, over a long period of time, which can then lead to type 2 diabetes, right, which we'll discuss in a future video. But anyway, the main things to know here is that we have 6 insulin for a zinc, and we have C peptide for every vesicle of insulin, all right? And the C peptide is going to come in later use when you have to see how much insulin you're producing in your blood, all right? So because we produce one C peptide for every insulin, that means our C peptide to insulin ratio is the same. So if we see the amount of C peptide produced or the amount of C peptide in the blood, we know that that's how much insulin's in the blood. All right, so it's good for blood tests, I guess you could say. All right, so now that we have insulin, right, and insulin's ready here, and we have many of these vesicles. So let's just draw many of these vesicles and represent 6 insulin to our zinc, right, all bound to our zinc. And then let's represent our C peptide, all right, and then same thing for here, all right, but just to save time, we know it's the same thing in these two. We have many vesicles ready, but how are they released? Now let's go back to here. When glucose enters in, right, then glucose can actually go and be converted into glucose 6-phosphate, right? Glucose 6-phosphate. But how though? Remember that in glycolysis we have an enzyme that does this pathway, this specific reaction. That enzyme's name is, it depends on the cell, right? If it's your muscle cell, it's going to be hexokinase. And if it's your liver cell, then it's going to be a glucokinase, right? Glucokinase. So either one of these enzymes will stimulate this reaction from glucose to glucose 6-phosphate. What does this mean? Didn't we use an ATP in this reaction, right? Remember in glycolysis? So what came out was an ADP and a phosphate. Remember, this GLUT2 transporter is bidirectional, so glucose can just as easily go out, right? And maybe not just as easily because there's such a high concentration here in the blood, but it can still go out because GLUT2 will allow it because it's bidirectional. How do we make it not go out? You see this phosphate that we used, right? You see this phosphate that we produce as a byproduct of using an ATP molecule. This phosphate will then go to the 6th carbon of glucose and attach itself. What will that result in? Glucose 6-phosphate. Glucose 6-phosphate now cannot go back through, right? So now it can't go back through because especially this phosphate group, usually charged compounds like the phosphate group can't really go back through a channel, or it can't enter straight through a phospholipid bilayer, all right? Sorry about that. But anyway, after this happens, we see that glucose, right, will continue through this glycolysis pathway. All right, so let's go ahead and scroll down here. So let's bring glucose, you know what? Let's bring glucose down. Oops, sorry about that. Random marks here. Let's bring glucose down here. And remember, guys, glycolysis again, all right? So glucose will then go to what? Glucose 6-phosphate, right? Just to save time, we won't mention the enzymes, all right? But we discussed actually glycolysis in detail, so make sure to go check those videos out for glycolysis. Then glucose 6-phosphate went to what? Fructose 6-phosphate, all right? And then fructose 6-phosphate went to fructose 1,6-bisphosphate, all right? So fructose 1,6-bisphosphate, all right? And then we're not going to write the whole pathway out, but we know that it'll continue to the rest of the glycolysis pathway. Then what we're going to do, right, at the end, what do we make? We make 2 pyruvates, right? Because glucose is a 6-carbon structure, pyruvate's a 3-carbon structure, so we split it into 2 3-carbons. So we make 2 pyruvates. Now, what will these 2 pyruvates do? Well, these 2 pyruvates, then remember we said will go to the mitochondria, all right, in particular the matrix of the mitochondria, and we'll do what then? We'll have the transition phase, right? The transition phase where we said that we make the 2 most important compounds is the 2-acetylcoenzymases, right? The 2-acetylcoenzymases, and we also remember make 2 NADHs as electron carriers that enter the malate aspartate shuttle through when we talk about the electron transport chain, and we'll talk about the malate aspartate shuttle in a future video, all right? But anyway, we make 2-acetylcoenzymases, and then remember, after the transition phase, guys, we go to the Krebs cycle. What do we do in the Krebs cycle? Well, in the Krebs cycle, we make some ATP through substrate level phosphorylation, but the main thing that we do in the Krebs cycle also is make our electron carriers, right? So our electron carriers. And then we take those electrons and those electron carriers into the electron transport chain on the cristae of the mitochondria. And what does that result in? Because of our complex 5 or ATP synthase, right, which we'll talk about in a future video, we're going to have a bunch of ATP production. All right, so now let's go ahead and pick up in the next video from here and talk about now what happens after we start making a lot of ATP inside our beta cell because of the excess amount of glucose that's coming in, right? Because of our carbohydrate-rich meal. So a lot of glucose is coming in, right? It's coming in, and then now there's a lot of cellular respiration going on, and we end up making a lot of what? ATP, and now we're getting closer to figure out what happens or how insulin is secreted out. We already said how it's synthesized through protein synthesis, right? It's a peptide hormone, especially in chromosome 11, where then we have some packaging in the ER and Golgi apparatus to proinsulin to this vesicle with our C-peptide and zinc, right? But now let's see what happens as the glucose keeps on coming in. We make more and more ATP, and we'll see how that acts on specific channels that are on the membrane of the beta cells. All right, so hopefully I see you guys in my next video.